polymers to go through. The next one is the most important of all of them, although I probably shouldn't say that, and proteins couldn't work without the other things, but proteins do most of the functions and most of the structures within a cell, which means within an entire organism. Proteins make up between 20 and 30% of the cell mass. They are structural, they are enzymes, they are contractile. They contain carbon, hydrogen, oxygen, nitrogen. Some of them have sulfur in them, and a few have phosphorus in them. They are polymers of an amino acid monomer that are held together with a bond called a peptide bond. And they have their specific shape and function due to 4 structural levels of organization. So we have structural proteins like collagen, found in all connected tissues, the most abundant protein in our body. Many of them have enzyme properties. Most of them, what we'll talk about, have enzyme properties. They are catalysts that speed up chemical reactions without being used in them. Disaccharide hydrolyze, proteases, oxidases, all kinds of enzymes we'll talk about all year. And some are there for transport, they're moving substance in the blood or across the plasma membrane. Hemoglobin is a good example. It transports oxygen, working with hydrogen ions. Contractile proteins in our muscles lead to movement. Some proteins are involved in communication. Others are involved in defensive mechanisms, such as our antibodies. All proteins are made from the 20 types of amino acids, the 20 common amino acids, and they're held together using a covalent bond between the carbon on one amino acid and the nitrogen on another, which is given a special name. It's called a peptide bond. Proteins have an amine group and an acidic group, so they have the ability to act as either an acid or a base. And the reason there are 20 different of them is because there are 20 different R groups that are attached to the basic structure of an amino acid. So here's the basic structure of an amino acid in orange. We've got a carbon attached to an acid group, a COOH, and an amine group, that's a nitrogen, 2 hydrogens. And then attached on the upper part, as it's shown here, of this amino acid is an R group. It might be as simple as another carbon, or it may be very complex. Those 20 different R groups, I went the wrong way, no I didn't. Our covalent bonds are held together by peptide bonds. 20 different R groups represent. So here's where we form the peptide bonds. So the OH from an acidic group of 1 combines with a hydrogen from the amine group of another, forming a bond between a carbon and a nitrogen, which gives us a peptide bond. So if we have 2 amino acids joined together, we have a dipeptide with a single peptide bond. The structural levels are primary, secondary, tertiary, and quaternary. Primary structure is the linear sequence of amino acids, the order of amino acids in the chain. There are some small areas of that chain that will spontaneously interact with each other to form secondary structures, such as alpha helixes, which look like a spring kind of, beta pleated sheets, which look like an accordion ribbon. But all of those secondary structures, and even places that don't have the secondary structures, will finally fold to give you a completely well-defined 3-dimensional shaped polypeptide, and that's the tertiary structure. If there are 2 or more polypeptides involved in making that protein or that enzyme, then we have to deal with the quaternary structure, which refers to how the different polypeptides interact with each other. So we can look at the primary structure, amino acid 1, 2, 3, 4, 5 in this case, and we could find that out by looking at the gene. The genetic code tells you which amino acid goes next to which amino acid. Doesn't give us a lot of information about the overall structure of the protein, however. But in that long chain, there will be regions that start to form these small areas of organization, like a helix or a sheet, beta sheet. And then as those helices, beta sheets, and other just linear portions work together, you get the tertiary structure, which gives you the overall three-dimensional shape of a single polypeptide. And if that single polypeptide works with other polypeptides, what that overall three-dimensional structure looks like is called the quaternary structure. And those levels of organization are very important. They give you the overall three-dimensional shape. They're caused by hydrogen bonding within the proteins themselves, and they are very much affected by things like pH. Let's see what the video can do for us. If we can get it started.
Most proteins are folded into a complex glob.
Whoops. Oh, try it again.
Most proteins are folded into a complex
globular structure. I'm going to try one more time, and then I'm just going to move on.
Most proteins are folded into a complex
Well, I promised you I'd move on. Nothing worse than an animation that's not animated. So, a lot of proteins are globular in shape, but don't think that globular means it doesn't have a definite shape. The other class of proteins are fibrous. They are generally structural proteins. They are more strand-like or rope-like. They are water insoluble. They are very stable over time. Most of them do interact with other proteins, so they have both tertiary and quaternary structure, very important three-dimensional structure. They're going to provide mechanical support, tensile strength. One we're going to see right off is keratin when we start talking about the skin, when we get to our first actual body system. We will also see elastin and collagen throughout the year. So the globular ones, though, are compact, spherical, water-soluble, sensitive to environmental changes. They're what I don't want you to think, though, is that globular means they're just a big blob. They're not just a big blob. They have tertiary and usually quaternary structure. Their three-dimensional shape overall might be described as a glob, but the three-dimensional shape is extremely important. They have functional regions on their three-dimensional shape, what we'll call active sites. And most of the proteins we talk about all year long will be these functional globular proteins, such as antibodies, hormones, chaperones, and all of the enzymes. Denaturation is a loss of that three-dimensional shape in a protein. Doesn't happen as much with fibrous proteins, but with globular proteins, the active sites can be deactivated. Denaturation can be caused by changes in pH, increased acidity, or increased temperature. In many cases, it's reversible if you put it back in normal condition, but that's not true for all proteins. We can get extreme temperature changes. You're not going to renature that protein. For example, the yolk of an egg, the egg, if you crack the egg and put it in a frying pan, that's mostly protein, and those proteins are in a liquid form. Once you cook it, it's a solid form, and you can't go back to an egg from a scrambled egg or fried egg. The big takeaway from this protein structure should be the enzymes. Those are biological catalysts. Catalysts are going to increase the speed of chemical reactions without getting used up in the process. They're going to lower the energy needed to initiate a chemical reaction. They're going to lower activation energy. That causes the reaction to go much faster, and we can get millions of reactions in a minute using these enzymes. Many of the genes, all of the genes in your nuclei, code for proteins, and many of those are enzymes that allow us to be alive. Most functional enzymes are referred to as holoenzymes because they consist of more than one polypeptide. There's the apoenzyme would be the protein portion. Cofactors we'll see as we go through discussions. Those are often metal ions, and coenzymes are organic molecules that carry things for us. Enzymes are very specific. They act on one specific substrate, and the name often ends in ASE for the reaction they catalyze. So a hydrolase will catalyze hydrolysis, an oxidase will catalyze oxidation. So they work by lowering activation energy, and that's the energy needed to get a reaction started. It primes the reaction. You can think of a rock at the top of a hill. If you give it a good push, it'll roll down the hill all by itself, but you need to give it a push. Well, enzymes help with that push. They help get things going. Substrates will bind to the enzyme's active site, forming an enzyme substrate complex. That complex will undergo rearrangement, giving you the product, and the product is released from the enzyme. If you think back to our chemical equations, enzymes bind to reactants and release products. And once they've released the product, they can go find more reactants. So here is activation energy on the left without the enzyme. It can be huge. With an enzyme, we can lower that activation energy to something that can occur within a living organism. For example, it takes 412 degrees Fahrenheit to cause sugar to burn, glucose to burn. We do it in our bodies at 37 degrees Fahrenheit because we have enzymes that allow it to happen, but it's the same chemical reaction. So substrates bind to the active site. The active site holds them in position while the bond is formed, and then the product is released. In this case, we're talking about putting two amino acids together. So the amino acids bind to the enzyme. Bond forms, water is released. Remember, it's a dehydration synthesis. And then when you have a dipeptide instead of two amino acids, the dipeptide is released from the enzyme. And that brings us to the end of proteins.